Hello everybody, this is Debadrita from Parul Academy. In today's video, we shall be learning about the different artificial methods of vegetative propagation. The artificial method of vegetative propagation includes certain techniques developed by humans to propagate the desired varieties of plants. Artificial propagation technique গুলো horticultural practice এ ব্যবহার করা হয়ে থাকে. The most common methods of artificial propagation are cutting, grafting, and layering. The latest techniques of artificial propagation includes hybridization, micropropagation, and biotechnological methods. Cutting. Cuttings are separated portions of root, stem, and leaves which are planted in moist soil for propagation. Cutting অনেক ধরনের হতে পারে, যেমন root cutting, stem cutting, and leaf cutting. For root cutting, a small piece of root is cut from the parent plant and put in the moist soil where they sprout, forming roots and shoot. It is found in lemon, orange, and tamarind plants. For stem cuttings, 1-year-old stems are used which show active growth. They are cut into around 20 to 30 centimeter long pieces and are dipped in root promoting hormones for few minutes and then planted in the soil. এই ধরনের কাটিং যখন মাটিতে পোতা হয়, এদের থেকে স্পেশাল রুট তৈরি হয় যাকে বলা হয় adventitious roots. The buds at the base develops into new plants. It is done in rose plant, sugarcane, hibiscus, and dhatura. For leaf cutting, the leaves are cut transversely into 2 or 3 parts and planted vertically in the soil one after the other. It is done in bryophyllum and agave. Layering. In layering, the growth of adventitious roots is induced on the soft stem, while this stem is still attached to the parent plant. যখন এই adventitious root গুলো তৈরি হয়ে যায়, তখন এই root সহ স্টেমের অংশকে কেটে নেওয়া হয় এবং আলাদা করে মাটিতে পুঁতে ফেলা হয়. And this ultimately grows into a new plant. The stem used in layering is defoliated and a small portion of the bark is cut open to expose the area and then cover it in moist moss, grass, soil, etc. The different types of layering includes air layering. In this method, the stem is girdled at nodal region. Girdling করার পর এই জায়গায় রুটিং হরমোনস দেওয়া হয় এবং ভেজা মাটি বা মস দিয়ে পলিথিন শীট দিয়ে বেঁধে দেওয়া হয়. Roots emerge in these branches after 2 to 4 months. Such branches are removed from the parent plant and grown in a separate pot or ground. এই টেকনিককে গুটিও বলা হয়. It is an ancient technique of propagation of tropical and subtropical trees and shrubs. Example, lychee, China rose. Simple layering. Here, soft basal partly injured branch is pegged at one place. Example, cherry and jasmine. Serpentine layering. Here, the branch is pegged at several places so as to form many plants. Example, clematis. Trench layering. The branch is pegged in a horizontal position. Example, walnut and mulberry. Mound layering. This method is applied for the plants having flexible branches. গাছের নিচের দিকে কোনো ডাল নিয়ে তাকে বাঁকিয়ে কিছুটা অংশ মাটি চাপা দেওয়া হয়। But the tip of the branch is exposed above the soil. After roots emerge from the part of the stem buried in the soil, a cut is made in the parent plant so that the buried part grows into a new plant. Grafting. The technique of joining of two different plant parts in such a manner that they unite and develop as a composite plant. The shoot system of one plant is called scion and the root system of the other plant is called stalk. The scion is a shoot about 4 to 12 inches in length. All the buds of the scion are kept intact while all the buds of the stalk are removed. The graft is placed on the stalk and the joining portion is covered with a layer of wax or clay to prevent evaporation and also the entry of injurious bacteria. It is practiced in plants like mango, apple, etc. The most common methods of grafting are splice grafting, whip grafting and approach grafting. আরো কিছু গ্রাফটিং মেথডস গুলি হলো টাং গ্রাফটিং, wedge grafting and crown grafting, bud grafting and side grafting also. In splice grafting, both graft and the stalk are cut across obliquely at about the same angle and then firmly tied together. Whip grafting is similar to splice grafting except that a tongue is added to the cut surface to provide better fitting and rigidity. It has maximum area of cambium in contact. It is common in fruit trees like apple, citrus, mangoes and also in rose and lemon. Approach grafting is also called inarching and is used to join together plants that are difficult to join. Here, the scion and stalk of independently rooted plants are grafted and the scion later is cut off from its original stalk. Micropropagation. This method includes propagation of plants by culturing the cells, tissues or organs. This is also known as tissue culture. For micropropagation, a small piece of tissue is taken from the growing tip from a plant and grown in nutrient medium under aseptic condition. The culturing results in formation of callus, which is an undifferentiated mass of cells which further differentiates to form large number of plantlets. These plantlets are separately grown to form mature plants. By this method, an indefinite number of plants can be obtained from a small mass of parental tissue. It is useful in obtaining virus-free plants, disease-free plants, homozygous diploids and quick commercial production of orchids, chrysanthemum, gladiolus and many more. Now it is question time.
What can it be? The undifferentiated mass of cells that may further differentiate to form large number of plantlets. The